Hello, everyone, and welcome to today's keynote presentation titled A Generic Manufacturing Platform for Downstream Processing on Non-mab Proteins. This webinar is part of the 4th Annual Bioprocessing Virtual Event, and I'm Susie Valdez of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots. So let's get started. Before we begin, I want to remind everyone that this event is interactive, and we encourage you to first participate by communicating with other attendees using our live chat feature during the presentation. You can find this live chat located at the right of your screen. You can also participate by submitting as many questions as you want during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. Now, if you have any trouble seeing or hearing this presentation, just simply click on the Help Desk button located at the bottom of your screen within the navigation bar or from the lobby. And finally, as a reminder, this presentation is educational and offers free continuing education credits. Click on the Continuing Education Credits link located at the abstract window below the presentation window and follow the process to obtain those credits. Without further ado, I now present today's speaker, Dr. Alois Jungbauer, a professor in the Department of Biotechnology at the University of Natural Resources and Life Sciences at the Institute of Bioprocess Science and Engineering in Vienna, Austria. For a complete biography of our speaker, please visit the Biography tab at the left of your screen. Dr. Jungbauer, welcome. You may now begin your presentation, sir.
Thank you for the introduction. I think everybody can see my slides. I guess so. Good morning, those who are actually in US, and good evening to the attendees here in Europe. I have seen even we have attendees from Austria. Yeah? So it will be a pleasure to give you an overview on the production and purification, mainly the downstream processing of non-mab proteins. Yeah? We all know that 60% of biopharmaceutical proteins are antibodies, yeah? But still there are a big fraction of proteins which are therapeutic proteins which are not antibodies. And they also have to be purified. The advantage of the antibodies is the generic downstream processing platform with protein A. I think I don't want to actually spend a lot of time on this protein A generic platform because it is so well known, yeah, and is so common and well accepted in industry. So we have worked in the past on actually a generic manufacturing platform, and while or generic manufacturing platforms, and then we came also across other platforms. I tried to give you an overview of a couple of these platforms, how they can be used for the production of non-mab proteins, discuss the pros and cons, and then I will give you also a couple of examples from our latest development. So the new, not only new therapeutic concepts, but a lot of therapeutic concepts are based on either enzymes, antibody fragments, peptides, cytokines, hormones, and also protein-based vaccines. Yeah? What they have in common in respect to bioprocess engineering is they suffer from a low developmentability and manufacturability. Yeah? And there is a need for a generic manufacturing and downstream processing. This is clear. Yeah? Compared to antibodies, we cannot actually be sure once we have overexpressed an antibody IgG that we can capture, purify the antibody with protein A affinity chromatography. So that's not possible. Yeah. So we actually have to find other ways. So I think the concept of fusion protein technology is actually not new. This has been pretty old. Maybe some of you have heard the name Matthias Uhlén. Yeah, he already started in the 60s, mid of the 60s. So a couple of decades ago, yeah, to overexpress proteins with a protein A tag and then purify it through an immunoglobulin G affinity chromatography. So this was the early start, yeah. So and others tried to tag proteins so that you have a purification tag. Yeah, when you see on the left, upper left on my slide, yeah, you have a purification tag, a linker, and then the target protein, or sometimes it is called the protein of interest, the BOI, the BOA. Yeah? So you could express them, then purify them with actually an affinity chromatography, which is specifically recognizing the tag. And afterwards, you have to cleave off the tag. Once you cleave off the tag, actually a small peptide remains on the N-terminus of the target protein or the protein of interest. If you have the tag on the C-terminus, the same would happen actually, that you have an elongated actually protein on the C-terminus. There was a big debate, there is still a big debate if we need an authentic N-terminus. So if we have to generate a protein which actually is cleaved really at the N-terminus. Yeah. Several groups over the past years, let's say decades, have tried to actually generate a generic expression system which relies on what you see on the right-hand side of this slide, where the protein is also tagged with a linker, but then the linker with the tag is cleaved off in a way that an authentic N-terminus is generated. So now let's go a little bit over. So this is very generic. This is a very generic procedure. It is very well established on laboratory scale, but actually rarely used in large-scale manufacturing. And one thing is, and this is the crucial point, this was always this cleavage of the tag and the linker. Yeah? So let's do the next. So the cleavage, what we could do is an enzymatic cleavage. We could do a decay, so this kind of quote unquote an autoprotease. The oldest way is the chemical cleavage, and then the inteins. And the inteins is also a kind of a chemical cleavage. Yeah, so it's not really, yeah. So that's the chemical cleavage were the first method really to actually cleave fusion tags from the protein of interest. Yeah? This is cyanogen bromide, actually cleaves at the methionine, so this is really, but then you have a lot of side reactions. This has been done in the past, and in this insulin processes, chemical cleavage have been used quite a lot. Yeah? But due to this actually a lot of side reactions, and you see you use a very hazardous chemical, this really has not become very popular. So there is one here, what you see. If you do like a fusion protein cleavage, you could, what I showed here already, at the first, you have this conventional tag, linker, target protein. then you would have tag, indane, target protein. Upon actually changing the redox potential, the tag would be cleaved off through the indane. Yeah, so because the indane decays, yeah, so that's or is disrupted. Yeah. The other thing is the autoprotease. So this is also a kind of decay. A lot of viral proteins are generated or expressed, yeah, as a long polypeptide. Yeah? And at the N-terminus, there is an autoprotease which actually chops up the viral protein into these different, let's say, proteins. Yeah? And this biological phenomenon can be used to generate actually a system for fusion tag cleavage. Yeah? So this autoprotease is overexpressed, yeah, quite often in form of inclusion bodies, non-native, yeah, and upon renaturation, yeah, there is a specific cleavage of the autoprotease from the target protein. So you see, we have used the autoprotease Npro a couple of years ago, and this is from the classical swine fever virus. Yeah? And you see, this function in nature is a proteolytic cleavage, yeah? Of the viral polyproteins. So these are non-structured proteins. Yeah? It is rather big, yeah, and we made a mutant of this Npro. Yeah? And this is quite interesting what you see here for this kind of decay when you do this Npro fusion. You see on the left-hand side the Npro, so in red with the target. Yeah? Overexpressed as inclusion bodies. And then upon refolding, the Npro is cleaved off and a target protein with the authentic N-terminus is liberated. On the right-hand side of my slide, yeah, you see the markers, the inclusion bodies, and the regeneration. And rather than see them in gray, where our target protein, in green is our actually fusion tag. So this is quite fascinating. The advantage of such a system is that what you see here already, that it is overexpresses quite a lot of protein, yeah, and the inclusion bodies can be washed and are already pretty pure, yeah. So the disadvantage is that you have to refold, yeah, and a lot of scientists don't like this refolding. Also, industry does not like to refold. Only a couple of companies really, let's say, fell in love with refolding of proteins. Yeah? So then you see there are another examples, yeah, what you could do. And the interesting thing is when you see here on the left-hand slide, so there will be the marker, then you have the induced and non-induced, the inclusion bodies, the renaturated, and then on this first STS page, you see the lane P, and this is precipitated. And this is quite interesting because the Npro is not soluble at acidic pH. Yeah, so you lower the pH to like pH 4 or so 4. Yeah, then the Npro is precipitating, and the target protein is in the supernatant. So this is a pretty simple, straightforward process. Yeah. So you see it also as two more examples on the second and the third slide, where actually we see with P1, P2, where we were able really to precipitate actually then the fusion tech and have a rather pure, you see, without any further chromatography, a rather pure undecked protein with an authentic N-terminus. Yeah. So then here you see some of these refolding strategies, yeah, and production strategies is the culture process, the centrifugation, the homogenization, the centrifugation. Then we would have the dissolution of the inclusion bodies into either urea or guanidine hydrochloride. Industry likes guanidine, likes urea, yeah. And then on the left-hand side, you would go for the refolding, and then the outer precipitation, and then you have a centrifugation, what I have showed you. Or you go to a refolding reactor and then do like a conventional purification by chromatography. So now let's go for the enzymatic cleavage. Yeah? And before I explain you about the enzymatic cleavage, let me talk a little bit about the properties or the specificity of enzymes. We have a protease, yeah, and the substrate, yeah. So the substrate actually, the protease recognizes certain amino acids. So this is the specificity, yeah. On the left-hand side, the amino acids after cleavage, yeah, it's called B1, B2, B3, B4, yeah. That's the substrate. And on the right-hand side is B1 prime, B2 prime, and so on. Yeah. So that's actually when you now wanna actually generate a protein with authentic N-terminus. Yeah. Your proteinase has to cleave off between the B1 and the B1 prime side. Yeah. Otherwise, you would not be able actually to generate an authentic N-terminus. But a lot of proteases are not specific enough. So let me review now a couple of them, yeah. So which are commercially available and used a lot in the lab. So you have the Arg-C proteinase, yeah. The Arg-C proteinase preferentially cleaves at arginine in position P1. Yeah. So if you have another amino acid in P1, yeah, you cannot use this Arg-C proteinase. Yeah. If there is another amino acid actually then is not useful, and also the P1 prime will interact and will moderate this cleavage. So then there is a lot of caspases have been used. So caspase 7, caspase 9, caspase 10. So these are enzymes which are overexpressed or induced upon apoptosis. Yeah. They are a little bit more useful for cleavage of fusion proteins. Yeah, what you see here, the caspase 7 has 4 amino acid recognition sites, and the P1 prime side is relatively flexible. Also the 7 and the 10. Yeah. So, but the problem of these caspases, they are very difficult to produce. and you cannot overexpress. You have to overexpress them in E. coli in a prokaryotic system because the caspases actually are induced upon cell death. Yeah, so they cannot be produced in eukaryotic production strains. A very common enzyme is the tobacco etch virus protease, the TEF protease. Yeah? Recognizes a linear epitope, which actually can accept quite a lot of substrates, yeah, and the cleavage occur between glutamine and glycine or glutamine and serine, yeah. And it is very common used in small scale, but not in industrial or preparative scale because the enzyme is terribly expensive. Yeah? So then we have the enterokinase. It's quite interesting. So recognizes a certain amino acid sequence, what you see here, with relatively high specificity. So here you got an insight by reviewing these different proteases. There are a lot more. Yeah. So I could talk an hour just on these proteases, which have been used for the actually cleavage of fusion proteins. But you get an insight how delicate it is and how difficult it is to generate an appropriate fusion protein system for large-scale manufacturing. Yeah. So this is the reason why this technology, although it is platformable, have not really entered industry. So it still remained with academia. So here you see only a couple of fusion tags. This could be peptides or proteins like the his tag, the C tags, a biopteam mimetic, polyhis tags, V5 tag, MYC tag, and so on. Hemagglutinin tag, yeah, and then proteins like the chitin binding protein, maltose binding protein, glutathione S transferase. Yeah? So for a technical purpose, yeah, you wanna use a small tag. Think about you have a big tag. Yeah, if you have a big tag, yeah, you sacrifice a lot of your metabolism of the cell to produce the tag and not the protein of interest. So over the metabolic, I'd say a lot of metabolic activity of the cells go into the production of tag and not the protein of interest. So this would be a waste of metabolic activity. Therefore, we want to have our fusion tag as small as possible. Yeah, so as small as possible. You could use or large tags have used for other purposes, either for increasing the solubility, for detection, or fusion proteins have been used a lot, yeah, for increasing the half-life of a protein. These are, for instance, the Fc fusions, yeah, to increase the half-life of a small protein, yeah. But this is not actually the subject of this talk here. So on the right-hand side of this graph, you see that the capture select, the C tag, the his tags, these are the smallest one, yeah, and they are also very commonly used in laboratory, in the laboratory. Yeah. So the advantage, what you see here, is with this is an example for the his tag. So this is overexpression of actually protein in E. coli. Yeah, and then you have two different type of chelates. Yeah, so because the histec itself, binds to chelated nickel, copper, cobalt, or zinc, and we can chelate this nickel, cobalt, copper, or zinc with what is called nitrilic triacetic acid, or with IDA. This is amino diacetic acid. And you see, here we have a lot of choices, and you always see on the left-hand side of like for the NTA is you have the supernatant, then the flow-through, the wash, and then the eluate fraction. Supernatant, wash, eluate fraction. Supernatant, wash, eluate fraction. And then you see with a lot of these chelated metal ions, in one step, a highly pure protein can be obtained. And we have a lot of choices of metal ions and chelators. In the lab, we use quite often a nickel, but a nickel is considered as carcinogen, and therefore for industrial scale manufacturing, this is not so well suited. So, and then I talked a lot about the authentic N-terminus, and you will question yourself, why is he talking so much about this authentic N-terminus? What is plotted here is the frequency of the N-terminus in the human proteome. So you see this on the left-hand side, so 18% is alanine, and 10% is serine, and then you see that. And on the right-hand side, you see the frequency of N-terminus in the human serum protein, proteome. And here you see glutamine, alanine, and so on. So these are different ones. And what you see here, the most of the tech removals are too unspecific, too expensive to produce this authentic N-terminus. And these affinity tags, to be together with his tag, can vastly simplify our purification. So what have we done? I already mentioned Caspases. We have used Caspase 2, and the Caspase 2 has the advantage that it has a recognition site of 5 amino acids. So, and think about when you have 5 amino acids, the selectivity will be will be increased, and they are independent of the B1 prime side, So, when you use now a linker with 5 amino acids actually linked to the B1 prime side, then this is ideal for the generation of a native N-terminus for fusion proteins. So we have what is called circular permutated, because this then allows a constitutive expression. Circular permutation doesn't mean anything else that we have linked together both chains of this heterodimer. So we have linked the C-terminus with the N-terminus, so that the protein is expressed as a single chain. So what you see here is this N-terminal authenticity. And this authenticity is quite often, this is expressed, the cleavage is K cut over Km. We measure this. Those of you who are not familiar with enzyme kinetics, so this is a parameter for the specificity, but also for the cleavage efficiency. So, and what you see here, with polar, non-polar, basic, and acidic proteins, we have different cleavage rates. When we bioprocess, then we actually are more interested in the time. And here we have plotted the same thing, but the time for 50% cleavage. So the time to cleave of a fusion protein, which an authentic N-terminus with glycine is less than 1 minute. And then we need a couple of hours for other acidic or for proline. We need even a day or more, and it is not really suitable. But what's the advantages of this enzyme? Is that it is can really highly overexpress. It can highly overexpress to a very high titer, so a couple of grams per liter, and therefore an enzyme is available for let's say industrial purposes, so that you can afford to use such a strategy. So the further advantage is that such an enzyme, and this is also really important, is stable at minus 20 degrees for 2 years, so this can be stored. And I think, moreover, this is more important for laboratory scale rather than industrial scale. You see here on the right-hand side, it is resistant to freeze thaw. So we checked 25 times, froze the enzyme, thawed it, and almost no loss in efficiency or in cleavage efficiency or enzyme activity. So how do we now proceed with such a generic platform for purification of non-mab proteins? So we have the affinity tagged fusion protein expression on the left-hand side. So the affinity tag, the red ones are the impurities, the green is the affinity tag. Then we do an affinity chromatography. In our case, it would be his tag. The tagged protein binds, the non-tagged, the impurities, they flow through. And then actually the tagged protein will be eluted, but it is also maybe a little bit co-eluted with other impurities. Then we add the enzyme, cleave off the tag, and do a subtractive affinity chromatography. The cleaved protein will flow through the affinity chromatography, and those proteins who are actually not cleaved will stick to this second affinity chromatography, and we get a native protein of interest pretty pure. So here I will show you an example. This is the case study for the production of human fibroblast growth factor 2, FGF. So you can produce FGF by other means, because it's a pretty acidic, pretty alkaline protein, but just as a model. So FGF2 was produced in E. coli with this N-terminal tag, and this consisted of these his tags for the affinity, a linker, and then this caspase 2 recognition site. So you see on the right-hand side, the his tag, the linker, and then the recognition site, for actually the enzyme to make it more specific. So here you see the advantage. This is expressed in a soluble protein with a relatively good titer, so that actually you get about 2 grams of protein per liter. Yeah, so that's a pretty good expression. Yeah. So now I will go through this slide to show you the purification of platform, yeah, for the downstream processing. So you have the marker with the left-hand side, the STS electrophoresis, the supernatant, and then we have the flow-through, the wash, yeah, and then we elute. And after elution, you see the cleavage. Then after the cleavage, you see the actually differential or the subtractive affinity chromatography, and you see actually a pretty pure protein. Yeah, overexpression is great, and a simple purification procedure. A final purity measured by reverse phase chromatography of 98%, 40 ppm whole cell protein, which is spectacular. Yeah, endotoxins, and then you see also rather low DNA impurities. Yeah. The major whole cell protein which has co-eluted is DNA K, and maybe have interacted with the metal chelate affinity chromatography. Yeah, I'm sure we could further reduce the endotoxins. Yeah, but we did not do the endotoxin, the second subtractive affinity chromatography with endotoxin-free water and so on. What you would do in case of pharma production. So here you see the upper slide that you already saw how this FGF has been constructed. Yeah, and then the panel C, you see the cleavage kinetics over time. Yeah, and then within 2 hours we have a complete cleavage. On the right hand side you see actually the cleavage kinetics, and then also the KCAT. Yeah, this is interesting for those who are actually interested in enzyme kinetics. So now let's go to the next. So the N-terminal authenticity. So it is fine, yeah, but we are not satisfied. Yeah. Here you see this KCAT over KM. I already showed you. So then what we did, we went to another selection round. So we selected, yeah, and you see here, this is the FGF. The example is 2 hours. Yeah, for a lot of other proteins left of the alanine could be processed much faster. But those right, there is a lot of this N-terminus which would suffer from actually a long cleavage. So we did then a further screening. I don't go into this selection system. You see here our recent publication, Profix, a bacterial selection system for the directed evolution of proteases. We published this last year in Journal of Biological Chemistry. Yeah. So we could improve the enzyme. Yeah, we increase the P1 promiscuity. Yeah, increase the activity, and have a similar manufacturability and stability as this original CASP2. So what you see here then, the non-dashed bars, see the improvement in KCAT over KM. Yeah. And on the very right-hand side with the proline, you already see that this proline can now be also cleaved pretty fast. Yeah. But again, what I have want to show you, because this Kcat over Km is more for science, and if you publish this, but more meaningful is again the time for 50% of cleavage. Yeah? So glycine was already 1 minute, so there was not a lot to improve. Yeah, but what you have seen here, that a lot of proteins now with different N-terminus can be cleaved within less than 1 hour, and actually the proline can be, when proline is the authentic N-terminus, can be cleaved also in a couple of hours. And now the system is really ready to be used for a lot of different proteins. Yeah? So here you see the system. Then we overexpressed also tumor necrosis factor with valine. Yeah. So these were the original. You see then on the left hand side. Yeah. The arrow points to actually the cleavage over time. The blue one is the original Caspase 2, circular permutated, and then the other one is ones are the cleavage kinetics of the improved Caspase. Yeah, so you see that we could substantially improve this, and everything what is below 1 hour is actually completely suited for industry. So I will pretty soon come to an end. So successful examples, what we have done so far, were fibroblast growth factor 2, tumor necrosis factor alpha, parathyroid hormone, human growth hormone, granulocytes, colony stimulating factor, superoxide dismutase, and nowadays everybody is working also on SARS-CoV-2. So we do the SARS-CoV-2 nucleoprotein, and with our collaboration partner Boehringer Ingelheim, we also overexpressed successfully 8 additional in-house proteins, but these are drug candidates, and they don't want to disclose the nature of these proteins. Yeah? And here you see we have recently published this also last year, the overexpression of the SARS-CoV-2 nucleoprotein, and this is already used in a diagnostic kit for SARS-CoV-2 antibodies. For the last part of my talk, actually for the Caspase 2, I want to acknowledge the team. Yeah. So the Institute of Bioprocessing Science and Engineering, the Institute for Molecular Modeling and Simulation, the Institute of Biochemistry from the University of Innsbruck, and Boehringer Ingelheim, the Pharma Bioprocess Science Austria. So you see, this is a huge team necessary to accomplish such a task. I hope I could give you an overview on the manufacturing of proteins, what are the challenges, yeah, and then our in-house example, which actually has successfully now transferred to industry, because this technology is now used for manufacturing of clinical phase material, not yet for full-scale manufacturing. And finally, part of this work has been financed by the Austrian Center of Industrial Biotechnology, which is a company owned by the Technical University of Graz, my university, the University of Innsbruck, and the University of Graz. Yeah. So now I think I'm the end of this my presentation, and I would be glad to actually answer questions or to get your feedback or comments to my presentation.
Thank you so much, Dr. Jungbauer, for that informative presentation. And we will now start the live Q&A portion of this webinar. If you have any questions you want to ask, please do so now. Just use that Ask a Question box located on the far left of your screen. So let's get started. Dr. Jungbauer, we have some great questions already coming in from our audience. Our first question: when do you want to express a protein as an inclusion body?
Inclusion bodies are not very well accepted in industry, but also not in academia. But sometimes you have a protein which is toxic to the cell. Yeah, so it's toxic. So it would actually reduce the growth of the cell, or it would even kill the cell. And then it's almost impossible to overexpress it. In such cases, inclusion bodies are a way out to get recombinant proteins in large amounts. So that's really why we are interested, or why people are interested still in inclusion bodies. Other reason why you want to overexpress in inclusion bodies, it's the high expression titer. Sometimes you would get up to 30 grams of protein per liter.
Thank you so much. And talking about overexpression, have you ever overexpressed viral proteins?
Yeah, you see, we have overexpressed the viral proteins, the nucleoprotein, but we have also overexpressed now proteins from Zika virus. Yeah, we have not yet fully purified them and characterized them. Yeah, so we have done this. Yeah.
Thank you so much, and I want to thank our audience for their questions today. Is there a rule for C negative or an N negative terminal tagging of fusion proteins, Dr. Jungbauer?
Quite often, proteins are tagged via the N-terminus. Yeah, so this is the most common one. Yeah, often the N-terminus may be hidden in the inside of the protein. Yeah. And therefore, a protease cannot cleave off the fusion tag. Yeah? So in such a case, you would go for a C-terminal fusion. Yeah? But it's also sometimes a matter of what you have established in your lab.
Thank you so much. We have time for a couple more questions. Is the fusion protein technology scalable?
What I have seen so far, yes, it is. It is scalable. Until recently, the fusion protein technology suffered from the high price of the enzymes for cleavage, if you want to go to a real large-scale operation.
Thank you very much. And we have time for one more question. Is it always necessary to generate a target protein with authentic N-terminus?
No, it's not. It's not. Yeah. Especially when we actually want to use a protein, yeah, for as a vaccine, yeah, then it is necessary, because we could get an aberrant immune response with the protein, where the N-terminus is not authentic. There are a couple of proteins, they need the full activity. For full activity, the authentic N-terminus will be required, but sometimes it is not necessary.
Dr. Jungbauer, I want to thank you for your presentation today. Do you have any final comments you'd like to make to our audience before we go?
What would be the final comments? Don't be afraid if you have to purify a protein which is a non-antibody. There are many ways actually to accomplish this task.
Well, I want to thank you again, Dr. Jungbauer, for your time today and for your important research. And before we go, I want to thank our audience for joining us as well and for their interesting questions. Questions we did not have time for today, and those submitted during the on-demand period will be addressed by our speaker via the contact information you provided at the time of registration. And this webcast can be viewed on demand for 2 years until April 6, 2024. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone. Bye-bye.
Okay.